Okay, so I think this is where we left off last time, isn't it? Talking about phylogenies, and we wrapped up talking about the bird being lumped with the crocodilians within the reptiles. Crazy, but that's what the DNA data shows. All right, so here's a phylogeny of our ancestry, Homo sapiens. Now, what we've done here is we have put a time scale on this axis. This is millions of years ago, right? And we've got the approximate durations of each of those species. So let's look up here, Homo sapiens. Homo sapiens have been around about, so humans as we know them, as you are today, probably about 40,000 years, maybe. But the species, and you looked a little different to what you do today if we go back maybe 0.25 million years ago, all right, is when the species first arose. And there's that fuzzy area we don't quite know. And there's Homo neanderthalensis, which died out not that long ago. But as you can see, according to this phylogeny, at some point, 1, 2, 3, 3 species in the genus Homo simultaneously existed on the planet, right? And more recent evidence has shown there might have been as many as 5 different species within the genus Homo that simultaneously existed on the planet. Now, they may not have all intermingled. They may not all have been in the same geographical area, just like you've got animals in Australia you've probably never physically seen, and if you had TV and books, you'd never know they were there. But they were simultaneously on the planet, which is pretty awesome. And then you can see what our phylogeny is. Okay, so look at that. A spin-off from Homo erectus gave to this species, and it's just awesome. And if we go back far enough to sort of the 4 to 6 million years ago, we get to see some of our ancient ancestors in this genus Australopithecus. All right, and so this species Australopithecus, we've got Afarensis. Can't read what that one is, but Afarensis is one of our oldest known human ancestors that was on a direct lineage to us. And we found some full fossilized skeletons of this guy in some caves in South Africa. And I think some more recent papers have shown that there's even something a bit more, a bit older than Australopithecus afarensis as one of our oldest known ancestors. But as you can see, other species spun off from Afarensis and they went extinct. They did not have ancestors that still persist today. All right. Okay, so let's see what we can do with phylogenies then, how we can apply them. Because there's a lot of different things we can do rather than piece together the evolutionary ancestry in groups. So we've used phylogenies, and this is one of many examples, to identify 2 species of wild grasses that are corn's closest living relatives. So think about this. Corn, as you know it today and see it in the grocery store, doesn't exist in nature. It's a result of artificial selection. It's what man has done to wild relatives of corn that resembled corn, but we bred them over years upon years upon years, and now we've got these big corn cobs, which, if we hadn't been around to do it, wouldn't have existed in nature. Does that seem surprising to some of you? In fact, take virtually every agriculturally grown plant species and virtually every agriculturally raised animal, and they don't really exist in nature. They're the result of artificial selection and breeding. So we've got corn then, which has ancestors out there in the real world that don't resemble corn. Now, what we would like to do is go out and find those ancestors, because when we grow corn, we need to have a wide genetic variety to choose from when we breed corn. For example, if a new disease evolves that kills corn, then we've got to go out into nature and find disease-resistant genes to breed into corn lines. So they're now resistant to those diseases. And we can only breed those into corn lines if we find ancestors, ones that are compatible with modern-day corn. Does that make sense? But with all the grass species out there, because corn is a grass, we don't know which are corn's ancestors, which ones we can breed with them. But by applying phylogenies, we can. We know exactly which species We're on a direct evolutionary trajectory to modern day corn. And I'll show you a graphic to represent that in a moment. So we've also used phylogenies to identify whale meat at markets that are illegally sold and illegally harvested. If you have a chunk of whale meat, sometimes you have no idea what whale species it came from. But applying phylogenies, you're able to match that piece of meat with a species, and then you know if it's legally or illegally harvested. And I'll show you a graphic of that. So here's modern day corn, and these are some of the relatives that are intermediates, and here's some of its ancestors. They sort of resemble corn, but not really. If you were to see these out in the real world, you wouldn't be able to tell if they were corn's direct ancestors or not. But using phylogenies, we can take samples, pull out the DNA, see which are the ancestors, and then we've got a gene pool that we can breed into modern day corn. So this graphic's in your textbook, and again it shows a phylogeny of all these different whale species: fin whales, blue, grey, humpback, minke, and then minke southern hemisphere whales. So what they did, I think there was a market in Japan, and they selected whale meat from the market. They got lots of different samples, and so you got sample 1B, sample 9, you can see all the sample numbers. And then they took them back to the lab, they extracted DNA from those whale meat samples, and they compared them in the same way that we did in lab with known databases. So with DNA from known fin whales, from known humpback whales, minke whales, and so on. And we were able to show that these unknown samples clustered with minke whales from the southern hemisphere. Those you can legally harvest. But then we've got these ones in red, so these clustered with fin whales, all right, either Mediterranean or Iceland. Well, you can't harvest fin whales. So we know that these were meat samples from these illegally harvested whales. This sample clustered with humpback whales, so we knew that was from a humpback whale, again illegally harvested, and this one from a North Atlantic minke whale, again illegal. Busted using phylogenies. It's pretty cool. All right, so constructing phylogenies then, we use DNA evidence, but we also use morphological data and biochemical data, such as enzymes. All right, so we use as much different information as possible in order to construct these phylogenies. It's not just DNA evidence. And of course, if you don't have tissue that contains DNA, you can't use DNA for your phylogenies anyway. So you're restricted to morphological data. So I want you to think about this. You know what phenotypic means, don't you? Phenotype, phenotypic. It means kind of the looks or something physical about an organism that you can measure. All right? It could be eye color. It could be blood type. All right? Something about the organism you can measure. So we've got phenotypic and genetic similarities among organisms, and they can occur as a result of shared ancestries. You share features with other mammals because you share an ancestry. And when your similarities in your phenotypes and your genotypes result from a shared ancestry, we call those similarities homologies. Okay? We call them homologous characteristics. You've got hair, lions have got hair. That's a homologous characteristic, because having hair is a result of a shared ancestry. Is everyone okay with that? I'm going to give you a bunch more examples. If you share morphologies, if you've got morphological similarities, and if you've got DNA similarities, then you're more likely to be closely related to each other than you are to species or organisms that have dissimilar morphologies or dissimilar DNA sequences. Is everyone okay with that? Similarities mean share, differences means not so much, at least not in recent evolutionary time. But when constructing these phylogenies, you've got to be careful because sometimes you'll get very similar morphological characteristics that are not the result of homology. Instead, they're the result of analogy. Now, I want you to be able to distinguish homology and homologous characteristics with analogy and analogous characteristics. And I'll give you a bunch of examples to illustrate it. So, homology results from a shared ancestry. Analogy, on the other hand, results from a process called convergent evolution. So, two organisms may share characteristics that look very, very similar, not because they share ancestors, but because they evolved in a similar environment. And as a result of evolving in that similar environment, they're likely to evolve similar characteristics. So I'll illustrate that with a couple of examples. So is everybody okay with convergent evolution then? Two organisms that might be very different to start off with evolve in a similar environment and as a result have similar characteristics because presumably those characteristics make them both well suited to that environment. So here's an example. This is the one that's in your book. If you go to Australia, you're going to find a mole-like creature that looks like this. And if you go to North America, you're going to find a mole-like creature that looks like this. Now, they look really similar, don't they? Yep, they've got very similar morphologies, similar characteristics. Look at these big, digging front limbs, streamlined body. Their eyesight is not great. Look at that sort of shape of the head as well. But they resemble each other, don't they, morphologically? But that's as a result of convergent evolution, not shared ancestry. In fact, this is a marsupial mammal, and this is a placental mammal. So evolutionary speaking, they're quite separate. Does that make sense? They're quite different. And the ancestors of these and the ancestors of these were also quite different. But because they're both subterranean burrowing animals, they evolved very similar characteristics. So these morphological similarities are the result of convergent evolution, and so they're analogous structures, not homologous structures. So do you see how you can be tricked? Just because they're similar doesn't mean they're homologous and came from shared ancestry. It could be that they came from convergent evolution. Good? All right. Here are some other examples of analogous structures and convergent evolution. Let's take a shark. And let's take a reptile, an extinct one like an ichthyosaur. And let's take a mammal like a dolphin. What were dolphins' ancestors? Cast your mind back to last week's lab. Antelope. Some kind of antelope. The ancestors of sharks were other fish. But look how similar they look. That's as a result of convergent evolution. Their ancestors were very different, did not share evolutionary lineages, but they both evolved in the ocean, so they both evolved these similar features. Now, superficially, they're similar. If we have a look at the internal structure of this fin of a dolphin, we find there are bones. If we have a look at the internal structure of this fin of a shark, it's a plate of cartilage. So again, you've got to be careful with these superficial versus deep resemblances. Okay? So, analogous structures from convergent evolution, not from a shared ancestry. On the other hand, similar with analogous structures and convergent evolution, I want you to think about the functional wing of a bat and a bird and a reptile. Not the underlying bone structures, but the functional wing. What's another animal that flies?
Insects.
Right, a lot of insects fly. Butterfly. bats, eagles, this pterosaur, and butterflies all have wings and can fly. Functionally, the wing arose from convergent evolution. Is everyone okay with that? Functionally, the wing is from convergent evolution, and it's an analogous structure. But if we have a look at the forelimbs of all these different mammal groups, there's the forelimb of a bird, forelimb of a bat, forelimb of a whale, forelimb of a cat, horse, and human. There's a huge resemblance in this underlying structure. So in black, we've got the humerus bone, and then this grey colour, we've got the ulnar and radius, and then the rest of them are phalanges, carpals, and metacarpals. So they all share this fundamental, although varied, underlying structure for their forelimb. That's the result. They are homologous structures, and it results from a shared ancestry. Is everybody okay with that? So it's interesting. The wing, functionally, the wing is an analogous structure, but its underlying structure of the bones, among a bird, an eagle, and a pterosaur, then they're homologous, those bone structures. And they result because they had an ancestor that had that very generalized forelimb bone structure. Good? Yeah.
So you have to look past the first appearance?
You do. And sometimes the first...
structure is what tells you that it's homologous?
In this case, the underlying bone structure are all homologous. They're all based exactly the same. But the functional wing is analogous. It evolved separately from different lineages. Okay? All right. So, bat and bird wings are homologous as forelimbs, but analogous as functional wings. Are you okay with that distinction? Okay. So, analogous structures, or even molecular sequences that evolved independently but are similar, we call those homoplasies. So homoplasies are similar structures or DNA sequences that evolved independently. They're similar because of evolutionary processes and not because of a shared ancestry. In the case of DNA sequences, the cause of these homoplasies, these similarities, usually is just chance. Random events can cause DNA sequences to become similar. Random mutation. All right? And I'll elaborate on that in a moment as well. Is everybody okay with this word homoplasy, though? Good? Okay. All right. So sometimes it's really difficult to distinguish homologous from analogous structures. But we can usually tease out what is homology by looking at lots of different lines of evidence. And usually, if you've got a very complex structure that are quite similar, when complex structures are quite similar, that's usually the result of homology. When it's more of a superficial resemblance, that's usually the result of analogy. Or when it's a simple structure that's similar, then it's the result of analogy. Okay? Right, so any questions then about homology, analogy, convergent evolution, homoplasies? Good? Okay. All right. So when we're looking at DNA evidence and we're looking at the similarity between DNA sequences, it's sometimes difficult, as you saw in lab, when you have a big, long DNA sequence, maybe it's 500 nucleotides long, and you've got it from 10 different species, and you've got to try and evaluate them, compare them for degrees of similarity. That can be tough when there are small changes in each. It's difficult to do it by hand, and it's difficult to do it by eyeballing the sequences. And I think that was one of the lab activities, to do that by hand. And those of you that did it found it was very laborious and labor-intensive and difficult to do. But now we've got sophisticated computer programs that can do it for you pretty quickly and pretty easily. So once we apply those computer programs, they usually are able to determine similarity in DNA sequences from homology versus analogy. Okay? Is everyone okay with that? Homologous DNA sequences arise from a shared ancestry. Analogous DNA sequences result from random mutations that just happen to make them quite similar. Good? Yeah. Analogous DNA sequences result from random mutations which just happen to make them fairly similar. It would be like this. If I took a deck of cards, shuffled them, and then turned over the first 12 cards, and I took that deck of cards, shuffled it, and did it again, there's a chance, random chance, it might be a long shot, that the first 12 cards would match the order, the second 12 would match the order of the first 12, just by random chance. So mutation, random mutation, can make things similar just by chance. Whereas analogous structures are from convergent evolution. Don't confuse the two. Good? Okay. All right. So, if we look at our Australian and North American moles, those are analogous structures, and we would expect the DNA sequences of these two organisms to be very different because the structures are analogous. There's no shared ancestry, so you wouldn't expect the DNA sequences to be very similar. On the other hand, the forelimb of a bat and the forelimb of a horse, their similarity is because of shared ancestry. They're homologous structures, and you would expect their DNA sequences to be very similar as a result. Good? Okay. All right. So you're okay with the structures, homologous and analogous. Now let's have a look at how we can get similarities in DNA sequences. So imagine here we've got the sequence from species 1 and we've got a short sequence from species 2. Now imagine these have just diverged from an ancestor. The divergent time is very recent. We would expect the sequences to be virtually identical, and in this case they are identical. Is everybody okay with that? You'd expect that, wouldn't you? As time goes on, however, by random mutation, you'd expect these sequences to change slightly, but independently of each other. So, for example, species 1, a random mutation might cause that G to be deleted. It's gone. And maybe with species 2, there might be a GTA insertion. Again, random chances cause that, okay, over time. So now we've got these two sequences. And if we were to compare them in alignment, we would say, wait a minute, these are quite different. But when we take into account it's just a deletion of the G and an addition of the GTA, computer programs can match them. Computer program says, all right, these are perfectly matched, but these two match those two. It's only because of that insert. And then the rest of those match the rest of those. It's just because of that deletion. Is everyone okay with that? So these, they bear a resemblance and similarity, but it's just a deletion and insertion which has caused them to appear superficially different. So a computer program would do that. It would align them like number 4. Good? And it would say, oh, these are quite similar. These organisms probably share an ancestry, and it's only because of that deletion and that insertion that they don't. So it's really important that we distinguish DNA sequence similarity. We're able to distinguish homology from analogy in molecular similarities. Okay? And again, the computer software we use helps us with that. So if you're a molecular systematist, or you use molecular systematics, you use DNA and other molecular data to let you enable you to construct these evolutionary relationships. Good? Okay. So what about this one? Here, we've got two sequences, and we can say they're similar there, they're similar there, they're the same there, they're the same there. All right? So this would be an example where these similarities are the result of random chance, not from a shared ancestry. And again, the computer programs are pretty good at telling you which is which, or at least they'll give you a degree of reliability. So we're okay with that? So now I want to jump to this topic of molecular clocks. What molecular clocks are and how they're used. So we use molecular data and the fossil record to piece together phylogenies. But we don't have fossil records of everything on the planet. Some things don't fossilize, some things are too old, some things we just don't have good fossil data for. But we can still use molecular data to piece together the relationships between them. All right. So I'm going to look at how we use this concept of molecular clocks to sort of calibrate when things happened. So I want you to think about a molecular clock. All right. And a molecular clock uses a constant rate of evolution of genes. In other words, it assumes a constant rate of change in DNA sequences. So let's think about this. If we've got a sequence that does, if we've got a sequence like this, and we assume that there's a constant rate of change by mutation. Let's just say one change happens, I'll just make something up, every 1,000 years. How many changes have we got? Not a trick question. 2. How many years do you think have passed? 2,000. It's as simple as that in concept. It doesn't work like that quite in practice. But so we assume a constant rate of evolution, a constant rate of change, and that allows us to calibrate the absolute length of time that's passed. So we look at the number of differences, and we say, all right, a certain number of years must have passed. So then we can compare two species or two groups. We can look at the amount that they differ and then estimate how far back in time that they diverged. Is everybody okay with that? You can see how that works? Yep. Do I have lots of nods or a bunch of shakes? All right, good. All right. So in order to calibrate the molecular clock, because different genes seem to, or different parts of your genome seem to evolve at different rates. I know we assume it's constant, but different parts evolve or change at different rates, although what we're assuming is that one part will have a constant rate of change. A different part will have a constant rate of change, even though the constants might be different to those different regions. Does that make sense? Just like you can have a car driving at 60 miles an hour and another one driving at 70. They're both going at a different constant speed. Okay? All right. So we usually need to calibrate the molecular clock using the fossil record. The fossil record we can date and we can put a time on reasonably reliably. All right? So if we use the fossil record to know when these two species split, and we know that it's a certain number of years ago, so on the fossil record we might say that's 60 million years ago, that time period, and then we can compare the DNA sequence data of a particular region between these two species, and let's just say, I'll make up a number, there are 50 changes, then we know that for that region, for those species, those 50 changes occurred over 60 million years. Then what we can do, we can go back in time, all right, and we can make inferences about when other groups diverge, because it's calibrated. Every 50 changes is 60 million years. Does that make sense? So maybe a species over here that has twice as many changes, maybe it was 120 million years ago that it diverged. Is everyone okay with that? Okay. All right, so different genes and parts of your genome do vary in how sort of clock-like they are. Some are quite clock-like, quite well behaved, and some change. So can anybody think of any problems with this sort of molecular clock? We're looking at the number of changes, and based on the number of changes, we can give an absolute time of divergence. What some problems may be?
The molecular clock is always changing. It's not something that will stay constant forever.
Well, so yeah, our assumption is, though, a particular region will behave in a reasonably constant way. It will change at a constant rate. A different region might have a different rate, but we assume that's constant through time. Okay? What could be some problems, though?
Natural disasters. I mean, because if they like wipe out
All right.
a lot of the population.
So we might have gaps. We don't have populations, we don't have enough, we don't have a complete DNA picture.
It's like chance events, like mutation. So like mutation could change how things happen over time. It could mess up the clock and the order we measure when things change.
So, but the changes in these sequences of the molecular clock, they are random mutations, right? So we do rely on that.
Random mutation that results in no change.
Or changing back, yeah. So think about this. We might have between these two 50 DNA differences, but it's possible by random chance. Do you remember the G deletion we saw? Maybe chance, a G gets inserted in that exact same spot, right? Does that make sense? So you can have back mutation, in which case we underestimate the number of changes. More changes occurred than what we've actually measured because mutations have gone back to the way they were. Yep. Okay. All right, so here's a graph then, all right, and it shows us the number of mutations against divergence time in millions of years. And so this is pretty good evidence of this molecular clock. Here we can show for a certain part of the genome that we've got a relatively constant rate of change over time. So 120 million years ago, we've got, what would that be? 60, 75 mutations at 120 million years. And then at 30 million years, we've got what looks like about 40 or 50 mutations for that region. So now we can look at the number of mutations and we know how long ago they diverged, or based on the differences, we can put a time to it. Does that make sense? But other regions might evolve at a different rate. So we need to know the speed of the clock for each region we want to look at. Okay? All right, so that's molecular clocks. Now we've got this neutral theory. Now, to explain the neutral theory, I want you to you can't do this really, but I want you to think about your genome as being two separate parts. It's not two separate parts, but we can think about the coding region and the non-coding. Non-coding region. So let's think about the coding region as being genes which code for proteins. And let's think about the non-coding region as being a part of your genome which doesn't code for any proteins. So if I was within a gene, do you remember the name of the parts of the gene that are coding and the parts that are non-coding? Do you remember that?
Are there introns and exons?
Good, introns and exons. So, I would just say this gene, this region here is an exon. This region here is an exon. This region here is an exon. That's the coding part. The introns are non-coding. And if you remember, when messenger RNA is made from these intron areas are removed from the messenger RNA before it goes out and gets translated. Do you remember that from 181? Yeah, maybe. Maybe it's a distant memory. Hopefully it's coming back. So, if we had change in this coding region, is that likely to affect the organism?
Yes.
Yeah, it could well affect it. It may not, but there's a reasonable chance it will. And most mutation is deleterious, bad. So what is natural selection going to do to mutations that occur in coding regions? What will happen by natural selection?
Typically will be selected against.
They'll be eliminated. Mutation that occur in the coding regions, if it's bad for you, if it negatively affects your fitness, natural selection will pull those out, will weed them out. Does that make sense? Good with that? Occasionally mutation has an advantage, in which case natural selection will make those more common in the population. Okay? What about if you get a mutation occur in the non-coding region? Does it have any impact on your biology or your fitness? No. So will that be affected by natural selection? Does it give you a survival advantage or disadvantage? No. So these non-coding regions are the ones that are really useful for our molecular clocks. The coding regions, well, natural selection changes them. It eliminates them, or it makes them more abundant. So it's really hard to calibrate a clock where the changes are either being eliminated or increased. Does that make sense? Changes in here is either going to make the clock go really fast or really slow. Change in the non-coding regions keeps it kind of regular and steady and clock-like because they persist. Natural selection doesn't change them. That's what the neutral theory states. Okay? Is everybody okay with the neutral theory then? Now, there are non-coding regions outside of your genes, of course. Yep. All right, so there are problems with the molecular clocks because they don't run as smoothly as this neutral theory would predict, unfortunately. Natural selection can change how the clock works, especially if you're looking at coding regions. And when we try to use or apply molecular clocks to organisms where there's no fossil record evidence, as we go further back than good fossils exist, then there's a lot of uncertainty. There's a lot of wiggle room. There's a lot of, you're not quite certain. Okay? There's a lot of unpredictability there. So what we try to do to improve accuracy is use lots of different genes or lots of different regions. Sometimes one region corroborates another region, and sometimes we can get an average of different regions. Is everyone okay with that? Good? Okay. So, let's apply molecular clocks then, as we've done, to the origin of the HIV virus. That's what we've done. So, of course, we've got HIV virus samples now, today, and we've got samples that were collected and stored from decades ago when the virus was first discovered. All right. Now what we find, which is quite kind of nice, is that the HIV virus, it descended from a virus that infected chimpanzees and other primates. All right, that's the first thing we know. based on DNA sequence similarity. We also know that it spread to humans more than once. That complicates things, right? But we also know that the HIV virus, its DNA, has changed in a very clockwise fashion. It's been very well behaved, very clock-like. So, when we apply the molecular clock theory and the neutral theory to one strain of the HIV virus, we can trace it back, and I'll show you a graph of how we did that. And we found that it made the jump to humans in roughly the 1930s from either a chimpanzee or some other primate. Okay? And here's the graph that showed that. So let's have a look at this graph then. So here we've got, so this is an index of base changes between HIV gene sequences. Right, so it's not absolute number of changes, but it's an index or a measure of the degree of change. Way down here, if you've got a value of 0, that says they're no different. And up here, a value of 0.2, that's a measure of change. It says these are very different. And here we've got year, a measure of time. So let's have a look at each of these data points. All right, so this would show an HIV it would show a sequence from a virus or a virus strain, and it gives you, so up here we've got an index measure of about 0.17. So let's have a look at all of these that were up in sort of roughly the year 2000. And then as we go back in time, we find that the number of differences decreases. That's what we would expect, wouldn't we? The longer the amount of time following divergence, more differences have accumulated. As we go back in time, we would expect the differences to be fewer. Is everyone okay with that? Now, we can't go back too far in time because I don't think we have the samples to measure it from. So what we do is we take these data points and we draw a line of best fit. And we zoom back to about the 1930s. And here's the sort of degree of unpredictability. Is everyone okay with that? How we've applied then the molecular clocks. So presumably, the HIV virus down here, there was maybe one strain. And of course, within that strain, there were no differences. Once it made the leap to human beings, over time, it's going to accumulate changes. Maybe a different strain would have a completely different line to it. I don't know. Yeah.
HIV was, you said it came, humans spread it more than once?
So it came from chimpanzees or some other primate. It made the species jump to humans, but more than one time. Okay? So that makes things a bit more complicated, right? Because Because let's just say here's one strain of HIV, right? And we can compare all our DNA sequence data among those strains. So we can say, all right, this is when it made the jump from one strain. And then we've got other samples of the DNA virus, and there are sequence differences, right? But there will be differences, but similarities. And based on those similarities, we can say, oh, this is a different strain. And it probably entered the human population at a different time based on the differences. Does that make sense? Okay. So recently, I guess over the past, probably the past 2, 3, maybe 4 decades, not that far back, definitely the past 2 decades, using molecular systematics, we've really shaken up and turned on its head taxonomy of life on Earth, how it's classified. So I don't want to date myself too much, but when I was in high school, here's what I was told. Here's how we classified all of life on Earth. I was told we had 2 kingdoms, the animal kingdom and the plant kingdom. And any species was either in one or the other kingdom. All right? That seemed crazy. We put bacteria in with the plants. We put fungi in with the plants. Basically, if it didn't seem animal-like, we put it in with the plants. And then we had little protists that were both plant and animal-like in their characteristics. And we didn't really know what to do with those. So we just said, we'll chuck them in with the plants. All right? So there was a lot of uncertainty back then. But as a result of molecular systematics, we've completely shaken it up. All right? And we've changed it into what it is today. So we've moved on from 2 kingdoms, animal kingdom and plant kingdom, to now 3 domains. And it's all based on this molecular systematics. What are the 3 domains? Eukarya, archaea, and bacteria. Within the eukarya, we've got 5 different... Oh, hang on. Sorry, I jumped the gun a little bit. Initially, we said we've got 2 kingdoms, plants and animals. Then we shook it up a little bit and we said, no, wait a minute, we need 5 kingdoms. The Monera, which are the bacteria, we lumped all bacteria in that group, the archaea and the bacteria because they're prokaryotes. The Protista, the plants, the fungi and the animals. That was a bit of a jump. It was a move forward. We said 5 kingdoms. Then as a result of the molecular systematics, we said, no, let's look at these 3 domains. So we added a taxon. And that's how we end up with the bacteria, archaea, and the eukarya, based on their evolutionary relatedness. Good? And we came up with that from this sequence data of lots of different parts of the genome. And here's what we found. The tree of life kind of looks like this. There's some common ancestor for all living organisms. And then we've got a branch off here into the bacteria, and then a branch for the archaea. Remember, these are those sort of ancient prokaryotes that are down these deep ocean vents. And here we've got the eukarya, all the organisms that you're probably more familiar with, the fungi, the animals, the plants, and the protists. Now, based on this molecular evidence, the archaea and the eukarya are more closely related to each other than they are to the bacteria. So there's the bacteria. You might be familiar with some of those, you may not be. I remember in 181 we looked at the cyanobacteria, didn't we? Remember? Maybe? Yeah. There's some of the archaea. These are the ones, again, that live in those extreme environments. And these are the organisms you're much more familiar with. So to get that tree of life, we've used largely rRNA genes. What does rRNA stand for? Ribosomal RNA. And so we've used those genes because they evolve very, very, very slowly. And we want to go very far back in time. So we've got to use regions of your genome that change very, very slowly. Why would these particular genes change very slowly? Why would your ribosomal RNA genes... Firstly, what do we use ribosomal RNA for?
Producing proteins.
They're involved in that. What does the rRNA literally form? What structures? Ribosomes. And it's on the ribosomes that we assemble proteins. So that ribosome, its structure is related to its function, and its structure is determined by the RNA, the sequence in the RNA. Any changes in the RNA might tweak its structure, and then it may not be able to perform its function properly. So changes in the ribosomal RNA are going to be eliminated by natural selection quickly and often. So beneficial changes are going to occur very infrequently, but probably very slowly over time. Does that make sense? Okay. All right. And then this has added a little bit of, I guess, fuzz to the picture. And that is horizontal gene transfer. So there's been the movement of DNA from one species to another, or even one domain to another. So DNA has been able to hop from one species or group to another for a variety of different mechanisms. Can you come up with a mechanism how DNA could swap, could be changed between one species and another, and interchange?
Not the one that's going to make everyone giggle.
What is it?
Sex. That's not one of them.
Right? But sex between what?
Different species?
Yes, different species. Sex between different species. That's going to cause DNA to jump from one species to another, right? As long as the offspring are viable, right? I don't think it was, well, I won't go there. So we've got this concept of horizontal gene transfer, right? It's the movement of genes from one genome to another, maybe from one species to another as a result of hybridization, or there are other ways. Can you think about another way where DNA might be able to jump from one lineage or species to another, from one genome to another? Viruses. Remember, viruses insert their DNA into their host. Sometimes they can pick up DNA from one species, pop it into another, and it stays there. All right? That can really muddy the water. And there's lots of different ways, other ways that we can move DNA from one species to another. You know what plasmids are? Extra bits of circular pieces of DNA that bacteria have. And bacteria freely move plasmids from one bacteria to another. Bacteria are very good at taking up DNA from outside the cell. Viruses do it. Plants, for example, have something called transposable genetic elements, what we call jumping genes, where the genes literally hop out of one cell and hop into another. So that really complicates things because we assume that a similarity is a result of shared ancestry. But you can get a genuine similarity if the DNA came from one genome into another by horizontal gene transfer. So DNA has made its way from organisms in the bacterial domain into the eukarya and ancestors of the eukarya. That's muddied the water a little bit because that would make us, I guess, unite bacteria and the eukarya more than what we should. All right, I'll show you another really good example of horizontal gene transfer that affects you. Who's this guy? Anybody got a grandpa or relative that looks like this? Maybe. It's Homo neanderthalensis. All right? Now I say this looks a little bit like my brother because my brother has these really big eyebrow ridges and sort of a little bit Neanderthal looking. All right? So this Homo neanderthalensis, extinct now, went extinct about 10, 20,000 years ago, but we have bones that have some DNA left inside the bones. We've been able to sequence Homo neanderthalensis DNA, at least some of it. And obviously we've got a complete sequence of Homo sapiens. Now here's what we find, and I included the link to the article which goes into this in more detail. Human beings, as the result of hybridization between the two species way back in time, Homo sapiens have between 1 and 4% Neanderthal DNA in your genome. In other words, 1 to 4% of your DNA got there from hybridization with another species, Homo neanderthalensis. And we've managed to sort of trace it back from interbreeding between the two species to about 60,000 years ago in Asia. That's when the gene transfer occurred. And we've been able to work out that all ethnic groups carry Neanderthal DNA, but that transfer occurred later in Africans. So that said, it probably first occurred in Asia, and then there was maybe movement back down into Africa after a lot of it occurred, and so that's why the Africans have a little bit less. So there are some interesting, I guess, ideas that have arisen from this. Now, one speculation is that autoimmune diseases might have a cause in Neanderthal DNA, you receiving Neanderthal DNA. So here's what autoimmune diseases do. They're diseases, very often genetically based, where your immune system attacks yourself. Okay? Now, your immune system is really, really good. Your immune system kind of goes around the body and it sort of touch bases with everything in your body, every cell. All right, so if I'm part of your immune system, I'm going to touch base with everything. All right, you're good. You're part of me, my species. You're good. You're part of me, my species. It's like a blind man going around touching the face of everybody saying, I recognize you, I recognize you, I recognize you. But the minute your immune system recognizes something foreign, like an invading bacterial cell, it says, oh, I don't recognize you. And then it pulls out its guns and it wants to shoot it. Right? That's kind of how it works. So now just imagine some of your immune system genes have a little bit of Neanderthal DNA in. They're expecting to find Neanderthal cells, but they don't. They find your cells. And they actually get a little bit confused because they're finding your cells with a little bit Neanderthal in. So they're going, all right, no, okay, I'm all right with you. You're kind of, yeah, you're mostly Homo Sapien. I don't know what you are. You should be Neanderthal or Neanderthal-like, but you're not. You're Homo Sapien-like. Then it starts to destroy it. And that's what they think might be the basis of some of these autoimmune diseases as a result of your Neanderthal DNA. It's only an idea, but it's a really interesting one. All right, so I think we're going to wrap up Chapter 26, Phylogenies there. Any questions before you'll shoot off? Don't forget lab. Don't forget your drawing books. Anything else you've got to remember? Online quizzes. Keep looking. They'll be posted. Right. Oh, one thing I forgot to tell you about. If you look on the Canvas website, I've opened a YouTube channel which will have all the recorded lectures on. Okay? So you just click on the link and you'll see the recorded lecture on YouTube.